Here it starts, it basically, I start with the end result at the beginning. And so this was the habitat of a purple membrane, purple bacterium, unfortunately not living up in the sky but in the mud. It doesn't have many photons, and here we see part of the cell in a very thin section, only 200 angstrom thick. With electron microscopy, you saw that many of these circular things, these are all chromatophores. That's the new word you have to learn today. And here you see a chromatophore. The chromatophore contains these proteins, 6 different proteins, and there are about 100 of them. And they are the inside, because it's a photosynthetic organelle, are many chromatophores, namely chlorophylls and carotenoids. And so now we are zooming in onto this chromatophore beast and rotate it around. So it's a spherical thing. The diameter is about 700 angstrom. And then we stop at a place where we just have all the key players at one place. We need the whole sphere to do things, but here we have it. And here's the main key player. You might have seen many of these green circles, and now I look at the main component of this sphere. It is called light harvesting complex 2, or LH2, and you see it contains many chlorophylls, 27, and 9 carotenoids. And they are the main light absorbers, but as you will see, as a first surprise, they are just not acting individually, these chromophores. Here we see a carotenoid absorbs sunlight, it gives it excitation to chlorophylls. And then, by the way, this is all serious science. Every atom is here really represented correctly rather than just Hollywood. And so here we have a chlorophyll absorbing light, and it goes then into this circular pattern of excitation here. This is called an exciton. So this is a quantum phenomenon. The electronic excitation is coherently shared. And now we want to see this protein in the context of the rest of the chromatophore, because it is interdigitation of processes that is really so exciting. And so now we want to see what one of these guys is doing to the rest of the system. And so we see what the proteins are made of. And now we just show the chlorophylls, because they are the first actors. And so we see now on the left side that a photon is absorbed by a chlorophyll, then they excite this exciton, this coherent ring, and then the next exciton is excited, and then the next one. So the language of transport are the excitons. They are doing the transport. Then we got figure 8 excited, the red one, and now the excitation ends up in what is called the reaction center for a good reason, because that's where the energy is converted for the first time from sun energy to electronic excitation, which is pretty trivial, to some first form of energy. So we see this particular system, an incredibly beautiful system here again. So we have in red the chlorophylls that form a figure 8 around 2 blue reaction centers. And so we will see then again how a photon is absorbed, and then it excites these figure 8 chlorophylls and forms an exciton, and then the exciton jumps its energy to the reaction center, in the center of the left blue, and there it is first spread out, but then it is focused at one pair of chlorophylls called the special pair. And so this is where the energy then is really collected, the sun energy, and now we want to have a closer look. And so here we see then the components that are doing the job, the substrates and the substrate and the factors. And here we see the local excitation that was populated at the end of this energy migration, and the result of it is an electron transfer. The energy is used for electron transfer to the substrate, the quinone molecule that gets now one electron. A proton combines with the electron, And now a second time this process happens, and the second proton is being added to the quinone. A second proton will come, and then we have formed a quinol. So we have made out of a quinone a quinol, and this quinol leaves the reaction center and adds energy then to what is called the Quinone-Quinol pool. That's where the first energy goes in this photosynthetic system. Actually also in our own bioenergetic respiratory based system, we also have exactly the same kind of Quinone-Quinol pool at work. And so now we want to continue what happens with the quinol, and so we turn this from outside in. We look now from the inside out and see now where the quinol is processed. So we are not happy just stopping, but we want to see the whole thing. So here we see the quinol coming from the reaction center to a protein called BC1 complex. This BC1 complex is even more amazing than the reaction center, very ubiquitous, and it contains quite a few components. And so here you see them. And so 8 cofactors and 4 substrates, it's a dimer. And so we now see what happens with that quinol that is loaded with electrons and protons. So there's one bound, QH2, you see it there. And now the electron will be taken away and the protons will be taken away from the quinol to turn into quinol. You see one electron goes down, no surprise. But the second electron is going up. It is like you have a plug in the wall and you can send the charge, one charge to the right side of the house and the other one to the left side of the house. Very difficult in physics, but here beautifully done. You saw also that the protons were shaved away. And now we still have the electron sitting there, and there comes a cytochrome C2 protein that is caught in the copy number 6 inside of the sphere, like 6 little slaves, or what I want to call them, that are taking electrons and bringing them where they came from. So we get a second quinol, and now the same thing happens. An electron goes down, and the second electron goes up. That's very, very crucial, this bifurcation. So it goes up. And now we also shed off the 2 protons. And now we have this funny thing up there. And the funny thing up there, QH2, is actually combined now with 2 protons. And there. And then what we got now is we used 2 quinols and we made 1 new quinol. It's like, one of these funny deals when you buy something nowadays. You have to pay $100 and then you get $50 back. They hope that you don't cash in. Here it does it. And so we are basically saving energy in turning the quinone-quinol pool energy into a proton gradient across the membrane. So amazing. Now these structures that are involved are so large that only today quantum chemists, and we are there belonging to them in some way, are capable of actually dealing with this kind of system. So now you see how the electrons are taken away from the BC1 complex back to the reaction center. So now everything is looped, nothing left over, everything is fresh, except we have pumped protons from the outside of the sphere to the inside of the sphere. And now these protons are being utilized by a system that you know very well, the ATP synthase. And so now we see the ATP. We have 2 copies of ATP in the system. And so we see now, it is a little bit probably of a gimmick for you now because most of you know how this actually works. We have a proton, we soak the searing of the ATPase with protons, and now we are then bringing a new proton and taking one out of the ring. The new proton comes from the inside of the sphere, and leaving one goes to the outside of the sphere. This process is only possible when the ring moves. The ring jitters, it's all thermal motion, but whenever there's a correct position, rotation forward, then the protons come from the inside and leave to the outside, give it a directional force. So now we are looking at the system from the side, and so you know that the same thing happening what you just saw before. I rotate the ATPase, and I do that mainly for the fact to show you that we can combine at the moment still from different species a whole structure of an ATPase, not just the C ring and the F1, but we have the whole cadoodle, and so we can really describe the entire system now, and so there is no atom lacking in that chromatophore system that I showed you. Everything is being simulated, and everything is correctly represented so that we can really, as biophysicists, deal with it. So now the energy of the ring goes up there. I'll show you later a little more about this, and we're getting then the ATP. That's finally the outcome of the sun energy being absorbed, and that's what the system is doing. It has all these processes interlocking in the chromatophore, and that is basically the new philosophy, that we want to look not just at the individual process. Of course we have to understand the individual processes, but we want to also understand how they work together. And there come many new aspects, and this togetherness is actually very important. We see here this scene that I showed on my front slide, that the components of this chromatophore work like a clockwork. They are thermal still, they work very precisely like a Swiss watch that I have on my hand. And you see now the processes that are interlocking. Of course, there are more processes, they're simplified: light harvesting, electron transfer, proton transfer, and ATP synthesis. And so the point is, we want to now go towards seeing the linkage as a theme of investigation, and not just the individual process. And there's a lot of fun and new things that you can learn.